Hello, everyone, to this session on neural mechanisms of psychedelic-induced altered states of consciousness, which is a collaborative work with Professor Catherine Praller and Professor Franz von Rieder at the University of Zurich and my PhD student, Devin Stalica. We are going to talk about psychedelics as a way to understand consciousness, or rather altered states of consciousness. So one can use these very powerful compounds to provide evidence for the normative theories, for example, free energy principle and active inference. And together with the generative models like dynamic causal modeling, one can use these in conjunction with the empirical paradigms like MRI, where one can measure the peak effects of these drugs and get very high resolution images, which can be then used to fit the generative models like dynamic causal modeling and provide understanding of the neural mechanisms of these altered states of consciousness. While this will be the main topic today, we all know that these drugs have a mounting evidence that they can be very useful. They can have a useful therapeutic benefits to treat illnesses like depression, substance use, and PTSD, together with the mindset and setting, which is another important aspect, which relates to how much dose is given, what are the environmental factors in which the dose is given, and the mindset which one has while one approaches the dosing session. So these are all important factors. We talk a bit about the brief history of psychedelic compounds, where the black boxes talk about the discoveries related to the classical hallucinogens, and the red boxes about the dissociative anesthetics. So towards the end of the 19th century, there was an isolation and identification of mescaline by Hafter, which was followed by the synthesis and discovery of LSD by Hoffman. There were then few influential studies using LSD. Then as they became popular, they appeared on the streets in 1960s, which led to the banning of these compounds. They were placed in the Schedule 1 in US, which made it impossible to do any research using these compounds. But then towards the end of the previous century, with the advances in the functional neuroimaging, there were a few more influential studies which showed with safety that these compounds can be used to treat. They can be very useful for treating some of the mental problems that we discussed, as I mentioned before. And so then there is now very, very much an excitement in this area due to the power that these drugs can provide to clinicians, to basic neuroscientists, to understand how the brain works and how to treat mental illnesses. It was initially pretty hard to quantify the subjective effects of these drugs. What I'm showing here is a validated instrument called five dimensions of altered states of consciousness, and results from a meta-analysis using LSD. What this results are showing here is that compared to placebo, which are here down, and the LSD effects are pretty much pronounced on this scale. So this is so there are 5 components of this scale, which are here, and their composite is here with altered states of consciousness. We see that there is a pretty big effect on one of the scales called oceanic boundlessness, and another one called visionary structuralization. And these two also have subscales. For example, oceanic boundlessness is driven by blissful state and the experience of unity, while visual restructuralization is due to changes, profound changes in synesthesia and in elementary and complex imagery. At the same time, we see there's not much change related to anxiety in one of the scales called anxious ego dissolution, which showed a bit more increase in disembodiment and insightfulness. Similar results are plotted here for psilocybin at 3 different doses, the brown, green, and red color curves. They show respectively the high, medium, and the low dose. And again, we see here that that with medium and high doses, there is quite a lot of effect here for the blissful state and also for visual alterations. So this is now widely used. These skills are now widely used in the experiments, and I'll show some of the results relating to these as well. Then one thing about their potential, the harm potential of these drugs. So this is a pretty eye-opening graphic which shows the histogram for which is basically every bar here is a composite of harms to others and harms to users. And as we see here, alcohol and tobacco and cannabis, they are pretty much at the other side of the spectrum compared to some of the psychedelic compounds like LSD and magic mushrooms. With a caution that these experiments with LSD and magic mushrooms, because of the legal requirements, are done in a very controlled research environment, which contributes to the results we see in terms of the safety. Okay, so now I turn my attention to generative models, change of tracks, and these are the framework that we use to understand the observed data. So usual way is to have these time series data from MRI and use these to calculate functional connectivity, which then can be used to look at how the two groups with and without drug, what are the changes happening in terms of the neural events that's happening. But here, what we have a generative model and a specific instance of it, what we call the dynamic causal model. As the name suggests, this generative model or the forward model means that we are more interested in how these observed data are generated by the causes, which we show here by the effective connectivity or theta. So a theta or the effective connectivity are thought of as the causes and the data that we observe as consequences. And this model, the forward model, which is basically about writing the likelihood model, represents how the causes map to the consequences. Once you have this forward model, you can use the observed data to do the model fitting procedures or the model inversion processes, and they result in the influence on the effective connectivity, which is hidden. So this is basically a very useful framework to look at neural mechanisms, and I'll show you specific examples of this. Here I also show some of the results when these sort of generative models are applied to the whole brain level. So here you see we have 7 very well-known resting state networks, which we then include in our model, and we do the model inversion, and we show these dynamic causal connectomes. These connectomes are different from usual functional connectomes because they do have the directionality and as well as the biological plausibility in terms of the heterogeneity of the edges in terms of excitatory and inhibitory connections. We then use these sophisticated modeling methods to combine it with the empirical paradigms with functional MRI. Yeah, we use these to test a very well established hypothesis, which is a thalamic filter model. The thalamic filter model talks about that the thalamus acts as a filter of information. So in usual conditions, you get a lot of sensory information. Thalamus then filters most of it, and only a little bit of information gets to the cortex, which it gets processed. And this is what we are showing here. Yeah, the hypothesis is this, that under psychedelics, for example, LSD or magic mushroom, the thalamus is more like a gatekeeper, which is no more gatekeeping, and there's a lot more information that goes into the cortex, which results in the overflow of information in the cortex that results in some of the subjective effects of psychedelics. We start to test this hypothesis using dynamic causal modeling. And the data we used from our collaborators, Catherine in France, a very innovative and sophisticated experimental design where every part this was given several conditions. For example, there's a placebo condition. There was a placebo given and then followed up with a moderate dose of LSD, or there was a pre-treatment with ketanthrin, which is a 5-HT2A antagonist, followed by an LSD administration. So using this model, we showed that there was in these chosen ROIs, there was an increased connectivity from thalamus to ventral striatum and from PCC to the ventral striatum, but while there was a decreased connectivity from ventral striatum to the thalamus and also from temporal areas to the thalamus. This is under when we compare the drug condition versus placebo, but when we compared this with the blockade condition, where we have the pre-treatment with ketanthrin and then the LSD administration, we see almost that this is more like inverse of what's happening here. There's not much happening in this direction, and this provides overall as an interpretation that there's a lot more information that's been going on into the cortex for information processing. And this was one of the first evidence that the in vivo evidence for the thalamic filter model. This led to this working hypothesis, which has been in works for a while. That's how the psychedelics induce changes in brain connectivity. So this goes like this, that these effects work through the corticostriato-thalamocortical loop. So this is what I was talking about in the previous slide. What's been shown with the evidence is this, that there's an increased functional connectivity between thalamus and the sensory cortical areas, and there was a decrease in connectivity between thalamus and the association cortex. Then, as I showed on the previous slide, that LSD also increases the effective connectivity between thalamus and the posterior cortex, and also the from the thalamus to the ventral striatum. Now, these overall effects that results in a reduction of thalamic filtering. Yeah, which is because there's an increased connectivity between sensory areas and the rest of the brain, which is not counterbalanced by the decreased integrated processing in the association cortex. This imbalance results in what we call the psychedelic experience. So this is a working hypothesis. One of the evidence is what I showed you on the previous slide. Then we also looked into what we call how these drugs might impact the hierarchical organization of larger scale brain modes. So this is a very well-known result from cognitive neuroscience that DMN, which is a task positive or a network which is related to internal processing, is anti-correlated with the task positive or those networks which are basically outward focused. So this is a functional connectivity pattern where, as you could see, there's a clear anti-correlation between DMN and the salience network and the dorsal attention network. Yeah, what we wanted that what's happening beneath. So this is what we observe as functional connectivity. What happens if we try to look at what actually happening in terms of effective connectivity as the causes of this. So what we showed is this, that there was so this is a very similar way to represent the matrices that I showed on the previous slide, but this time there's a directionality and asymmetry in these matrices because these are effective connectivity. What we show is this, that all the regions that belong to the same brain region, they had very strong connectivity within that brain network. Yeah, and then what we showed is this, that the task positive modes, for example, the salience and the dorsal attention network, they inhibited the default mode network. And on the other hand, the default mode network had an excitatory impact on the task positive networks. And this is what we, at the level of regional effective connectivity, when we collapse this into a network level connectivity, we showed that the CNS network sits at the apex of this network and controls what's happening between default mode network and growth retention network. Now, with this, what we wanted then next is to see how this network organization changes with the ingestion of psychedelic compounds. So this is again a result from the same experiment where 2 resting state scans were taken after a placebo, one after 75 minutes of ingestion of LSD of 100 microgram, and another resting state scan after the 300 minutes. Here, the idea is this, that because one of the main subjective effect of LSA would be the ego dissolution, which we thought of as a blurring of boundary between the object and the subjective world. So what we show here is this, that there is an increased connectivity and impact of salience network on DMN, and there is at the peak effects. This is the mean, the medium, the middle one here is the peak effects, and these are the later effects. And the peak effects we see is this, that apart from salience network having a large impact on DMN, there is also an impact of DMN on dose retention network, and this goes down again, not to the same level as placebo, but very near to that. So this is again shown here on this graph, which is the same data plotted differently, and here we show that the same effect at the post 75 minute. There is an increase in effective connectivity, and then sort of coming back to where we started at the 300 minutes. When we look at the regional level connectivity, we see quite a lot of similar at regional level effect. And we also show here the behavioral association with ego dissolution scores from the 5-day ASC module, where we associated them with the effective connectivity. What we show here is this, that the salience network connectivity may contribute to the shift in the minimal south, which is defined as, which is basically an interpretation that we attach to the salience network, and we also see a shift in the native self, which we associate with the default mode network. So this contributes to the evidence about the blurring of boundaries between the subject and object world. So this is another experimental evidence of how the subjective effects are coming about and what are the underlying neural mechanisms. I would like to now lastly mention one of the clinical trials that we are doing currently in our lab, which is actually the first Australian trial. We call it PsyConnect, a study in which psilocybin will be given to healthy participants. In this study, we have 60 people. There are 2 groups. One group does 8 weeks of meditation. The second group doesn't do any meditation. And then we have 2 time points that both groups are imaged without psilocybin. This is what we call the baseline imaging, and then they are again imaged after the psilocybin as ingestion at the peak effects, and we image them with many different modalities, including MRI and EEG. And within MRI, we also have naturalistic paradigms that the data that we collect for. This study is, as I speak, currently ongoing. It would finish in October 2022. So with that, I would like to thank lots of people, my collaborators at University here, my PhD students in the computational neuroscience lab, and lots of other collaborators at UCL, Monash, and also many of the international collaborators, along with the sponsors who have made this work possible. Thank you very much.